cobble,
all good,
boulder, and rock, ridge. And each one of those has a size category, you know, between 6, it's pretty specific, it's quite funny, down to fractions of millimeters, the difference between sand and gravel.
Would y'all like to hear a question from New Zealand?
Sure.
Okay, it's a very logistic forward question. So here's the question. When you collect specimens, what is the collector name you put on the label? Is it the name of the operator of the arm or the expedition name? That's the question.
As far as I can recall, I think it gets the expedition name. It's EV Nautilus and then the dive, the specific dive that it was collected on. And then it usually also includes notes about having been collected by the Hercules. Usually they want to know what vehicle it was collected with. If people would like to see an example of what those labels look like, all of the specimens collected by Nautilus go to the Museum of Comparative Zoology at Harvard, and they have a publicly searchable catalog. And if you put in EV Nautilus as one of the search terms, it'll come up with a list of all the samples that have been generated over various expeditions, and you can see what each of those labels and what kind of information gets collected with each sample is.
So even our friend in New Zealand who asked that question can go see that, which is pretty cool.
Yeah.
Thank you. We also have a question about pollution, and this person is asking, is there much, is pollution seen down there? I'm paraphrasing their question, but does anyone want to speak to that?
Well, we do occasionally see marine debris as we're doing these explorations. We try to quantify it and keep track of different types and how much and where we find it. But I know the Trawl Survey collects that information too, so we're trying to get a better picture of what the debris load is on the West Coast.
So far, in the last couple dives I've watched, we've seen some glass bottles. We've seen leftover fishing gear in a few places, and some pieces of plastic. Got a pineapple siphonophore in the lower right-hand corner. The siphonophore that's benthic hovers right above the seafloor and uses its tentacles to feed on things that it finds on the seafloor as opposed to being up in the water column. And siphonophores are related to some of the jellyfish and in those families. An octopus. The deep sea octopus. We're getting less cobbly now and more into just like really more kind of bedrock looking formations. I was about to get excited that there was a coral, but it was a crinoid taking me out on the right.
We've got about half an hour to get one.
Yep. What will happen is what always happens, which is we will do watch change, and then they'll get into a beautiful coral garden. And then Kurt and Jen will get to narrate it all.
Does happen in reverse sometimes.
I'm getting an endless supply of questions, so I have this job to pick which ones I want to know the answer of too. And I have this question with our friend who asked a question a minute and a half ago. And they ask, why do fish at this depth have such a different shape from those at shallower depth?
Erica, you want to field this one?
One of the reasons is the pressures at this depth are pretty incredible. So being more jelly-like and having a thinner body shape helps fish handle that pressure. It's very dark and very cold down here, so some of the adaptations that fish have are larger eyes to collect any possible light, even bioluminescence from other organisms down here. What else? One of the things that the grenadier have is oil in their swim bladder instead of air to deal with the pressure. It fills up that airspace with something solid, and I believe the ROVs use that technique too. A lot of their pressure housings are oil filled to deal with the extreme pressures at these depths.
Yeah, 40 meters 110. Thank you. Yeah, I had heard that the like eel-like, what do they call it, anguilliform shape is also just like super efficient swimming-wise. So you get a lot of those type of, like a body that's more modified towards that type of sinusoidal swimming rather than big pectoral fins and stuff like that.
Makes sense.
Yeah.
Yep. In these habitats, conserving energy is often the name of the game. Speaking of which, we've got a big eel, or at the sculpin, just going off the bottom of the screen. Blob sculpin.
See it in the bottom.
Oh, those are super cool.
So we're kind of on this flatter part already. Looks like our map is matching up pretty well with our sensors. So we're starting to mosey a little to the south over this kind of flatter part. Looks like the slopier part was the rubble. Now we're getting some consolidated boulders and stuff.
Well, if we're starting to head to the south, why don't we go ahead and set up and start our first transect, since we're headed towards that next waypoint.
Okay, Roger. I may change our heading a little.
Okay.
Is
Whenever
that
you are
octopus?
I think it was.
On the left?
It was just wedged up against the wall. I think it was another octopus. And there might be one coming up here on the right.
Before I transect you? Sure.
Yeah, we can, you guys all set up for that?
Negative.
Okay.
So what's you thinking for the heading for the? Mhm. Um, 130 works because then we can stay along the depth contour. It's usually groovier for us. Right. Roger, yeah. So we'll do a transect at 130, Kim. Oh, yeah, maybe my mic needs to be closer to my mouth there.
I can turn Jess up.
Yeah, that'd be great, Marley. Thank you. Then you can go ahead and call it in since we're pretty deep.
I have a question that might take some brainstorming from someone who asked 2 minutes ago. So I want y'all to think about this. The question is, what is your favorite sea organism that you have encountered on the Nautilus? Take your time. You don't have to answer now, but when you think of it, why don't you share it?
So, yeah, we're just getting in position to start the transect. We're going to keep along this contour, not try to change our depth too much. So we're almost ready for that. If you,
Yep, stand by one.
yeah. And my answer to that is the Pelecanoides gulper eel that I saw my first year out on Nautilus 3 years ago, which was just very exciting. Very beastly creature that took a while to figure out what it was actually doing. And that was pretty neat. Satisfied the kid in me.
Yeah, I knew my response immediately too. It's the Iridogorgia octocoral, which was the very first thing I ever got to film on Nautilus when we were going from Samoa to Hawaii last year. And it's a beautiful spiral-shaped coral. If you Google it, I think our footage from Nautilus is the first thing that comes up. It's just really, really beautiful.
Nice. They have those at Davidson, and I'm really hoping we get to see one in the next couple days.
Yes, that would be great.
I've got to hold the one at the Smithsonian that came off of Davidson from years
All right, then
and
go ahead and
years
start
ago.
it.
All right, so we're going to mark this the start of the transect. Roger.
All right, stepping forward.
I have to say my favorites are anything found this deep with glowing eyes and big gnarly teeth. Viperfish, anglerfish.
Yes.
Any of the uglies.
Yes, the beast. Big fan of that.
So I'm going to have to go with 2016 when we were off of Oregon and we went to a big just garden of bamboo corals. And they were these big meter-high candelabras and they were just everywhere.
pretty fun. Folks from home are saying what their favorite thing is that they've seen on these dives, which is really fun. Someone said a sperm whale and whale bones from last year. So we have some long-time followers and fans tuning in too. Pretty cool.
Do you see we have the question, are creatures' eyes hurt by the bright lights from the ROV? And we don't really have any evidence that they're hurt. You will see some, sometimes you'll see fish that are unused to this bright light and they'll get a little disoriented. And sometimes they might even swim into the seafloor. But that seems to be just a temporary passing effect. One thing we do watch out for is you can have changes in fish behavior with the lights. So a lot of surveys have done some work trying to do work with just quick camera flashes versus continuous lights to see how fish behavior changes in response to the lights of a vehicle like the Hercules.
And just to confirm, we're still targeting 200 meters for the transects?
Yes,
Okay,
yeah.
got it. Yeah, some in situ behavior I imagine being pretty hard to study with an ROV in the larger, more kind of responsive stuff. Like the octopus always, you know, flare out their tentacles at us defensively. Hopefully they get back to normal pretty quick.
Yeah, in some areas that are shallower, you'll actually get krill and some of the more kind of prey organisms that are attracted by the lights, and they will in turn bring in other fish that will end up using the ROV to hunt. We've had several dives where we've ended up just completely surrounded by rockfish who are using the ROV lights to bring in the krill and then hunt and feed on the seafloor.
Our own micro migrations.
Yeah.
Look, we've
We found the soft bottom.
Yeah. Be easy counting during this part of the transect.
Yep. So for those listening along at home, we're doing what's called a quantitative transect. So we're having the pilots fly at a fixed altitude above the seafloor as much as they can maintain it, and at a fixed speed. And this video footage will be sent to NOAA's Southwest Fisheries Science Center, where they'll review it and they will count all of the fish and all of the invertebrates that they see that are over 10 centimeters. And they use those lasers as the reference point for 10 centimeters, and they identify them as species. And we do this up and down the coast using these same methods, and that allows us to compare between these different habitats and characterize the differences between them.
We've actually had several questions really related to the crew's kind of professional development, which I think is really interesting. We've had 3 questions about this. So if there's time, if you feel like being introspective, we do have questions about, was there a pivotal moment in your life that you decided you wanted to do science and marine biology? And questions like, what did you major in college? So if there's some downtime, there are people asking about that.
I think we can answer some of those as we're flying along in the mud part of the transect, certainly. This is Meredith. I've been interested in marine science since I was a very small child. And so when I was in high school, I decided I really wanted to do marine science and applied only to colleges that had marine science programs. I went in thinking I was going to do marine biology, more kind of looking at ecology and coral reefs and coastal environments, and ended up getting really into oceanography and working on planktonic systems and then getting into genetics. So it's been kind of a winding path, but always being interested in the marine field. And if you're interested in doing that, I would say go in, but go with an open mind and be prepared to like try new branches of the science. Now that I'm doing marine biology professionally, I'm really interested in the marine engineering side of it. How do you build new instruments to sample the biology?
You're right, Meredith. There's so many different ways to either specify or narrow down your focus or broaden it and study an entire ocean basin. Or you can study the bacteria that grow on certain crabs in certain areas. It's whatever you want to make of it, for sure.
Oh, I think we're seeing some more rocks. Kurt has just joined us in the science chat, and he has brought the rocks back, apparently. I can say to Tom Lighting, who's not actually with us right now, that I promise I don't try to put transects in mud on purpose so that I can sample the coral. I do actually want him to count the coral. Tom Lighting is our biologist at the Southwest Fisheries Science Center, who, along with Di Waters, who will be standing some watches during this cruise as well, do a lot of the video analysis with their team of this video we're recording right now.
Could probably knock it out in real time for this one.
Yeah, I think this one will be very easy counting for them.
But that's data. Zeros.
Yeah.
Rich.
Are we still doing pivotal moment in career stories?
Yes, please.
Sure.
All right.
Yes.
I always like to share mine just because mine's a bit different from a lot of other people who come on Nautilus. I don't actually have any sort of science background, which is important for some people to hear who are really interested in doing this type of work, but maybe are not scientists. My degree is in journalism, and my 10 plus years of my career so far has been centered around photography and video production. And I specifically started working with marine scientists more, well, my brief short story version is I was on a boat with a couple marine biologists back in 2014 who were releasing some rehabilitated sea turtles that had washed up on shore, and we took them out to the Gulf Stream to put them back into the ocean after they had been rehabilitated at some local North Carolina aquariums. And I got to hold one of these little Kemp Ridley sea turtles and actually put it back into the ocean and watch it swim away. And that was the day I decided I really loved working with marine science people. And they said I should get certified to scuba dive so that I could do some of this stuff underwater. And the rest is kind of history as far as my career with working with marine scientists and oceanographers on all kinds of field work and expeditions around the world. So get on a boat. Doesn't have to be the Nautilus, just any boat could you know, inspire you to pursue something.
I just saw a small black coral about a minute ago on the same rock that one of those white sea stars was on. So they are here, and hopefully we'll see some more. We just have another octopus that just went under the lasers as well.
Cutie.
Tiny.
Mm-hmm.
Meredith, did you write down the start time of this by chance? My computer clock is off.
I had 0231 as the start.
What do you have, Mary? Because I wrote my target, I dropped at 0236. So I don't know.
0236. Okay.
Okay.
I
Maybe
put
the
it in the
data.
chat at 0237, but let me,
Yeah.
I put it
I
in
think
the
we were
event
setting
log
up for a little. Yeah.
a little before that though.
Got it.
I
Yeah, we
put
were
it in our notes now as 0236, so it corresponds to the target.
great.
I have a question to test your memory and maybe creativity. There's a question that says, what was the sponge-like thing you collected yesterday? It was white with black dots popped out.
I couldn't quite copy that. What
Yeah.
was the question again?
What was the sponge?
What was the sponge-like thing you collected yesterday? It was white with black dots.
Is that the moldy cheese sponge?
Moldy
cheese. Is that what they were
The
talking
moldy
about?
cheese, yeah. It was a sponge they were actually looking for actively. You know, they have never collected it, but it was this, they called it semi-infaunal, which means it's living below the sediments. Yeah, but it was really small. It was hard to collect for
It
Jess.
was, in the lab we measured 2.5 centimeters,
Oh,
I
really?
believe. It was very tiny.
2.5
Yeah,
centimeters.
Baby.
They're really small and really hard to collect.
Jess did a good job.
Team effort. It was one of those classic moments of seeing it on the video screen and thinking it was like roughly the size of a ball, and then watching it come off the ROV, it was tiny.
Yeah.
A marble.
And it could have been some of the other sponges. We collected a lot of sponges yesterday. It was a very sponge collection heavy dive, which is good because sponges are at this point very poorly understood, and they're a major target for research right now for the deep sea coral program. But yeah. So.
This is an important question. It is, what is the current depth and water temperature? See where folks can find that information.
Depth is 1521 meters. Water temperature, 2.6 degrees.
And I think on Nautilus Live, if you're watching through the Nautilus Live website, if you scroll down on the right-hand side, they have been displaying the Herc depth in real time. Yep, it's on the right-hand side of the feed. So if you look if you're on nautiluslive.org, if you look on the right-hand side of the screen, it gives the depth in meters and feet. So we're also at 4,990 feet, and a water temperature 2.7 degrees C or 36 degrees Fahrenheit, so just above freezing. Abby in the science chat says, huge thank you to everyone on Nautilus who's processing all these tiny samples as they come up.
Yeah, Mary. Get it. You guys had a lot of sponges yesterday.
Lots and lots of sponges.
Another little baby octopus.
Oh, we've got another octopus. Just went off the bottom of the screen.
Are you familiar with that species? Is the adult small size like that?
I'm not that familiar with the deep sea octopus.
Looks like this. What did they call it? The googly-eyed squid? I don't know. Some Nautilus highlight. Or maybe it's a squid. Yeah.
Okay, we have a math question. Are we ready? Here we go. I'm going to assume yes. So the question is, how do the lasers maintain a fixed distance? Does it adjust angle, or are they just very
ROV switching out.
precisely aimed?
Lasers are parallel. So one of the features geometrically of parallel lines is they will stay the same distance apart no matter where they're pointed. And so that's what makes them a powerful tool, is it doesn't matter how far away they are from the object or the seafloor. If you can see those lasers, they will always be 10 centimeters apart, and they move with the camera, so they'll stay in the field of view. And if it gets quiet right now, I think we're at a watch change, and we may have our next set of pilots coming up into the van on the ship. So it'll go quiet for a little while here as the teams change out.
Yo.
Video is switching over.
Thank you all. I'm going to jump off so that Kurt can get on. He has 5-3 today. So Erica, I will turn him over to you to fill in about what we're doing so far. I did just tell him in the science chat that we are still on transect. But I will see you all later. Thank you.
Thanks, Meredith. I was typing in the same message, so I will fill him in.
Hey, Erica, it's Kurt. Can you hear me?
Yeah, we can hear you. Just so you guys know,
Great.
I'm going to change out, but we have about 30 meters left in this transect.
Great. Thanks, Ben. Sounds good. And this was transect number 1 for the dive. You're breaking up a little bit, Erica. Got it. Thank you. I have the transect tracking sheet, and I think Jen's going to cover the data recovery report. So yes, we have that. Okay, sounds good. And did Meredith get a Niskin already? Yes.
Alrighty. Hello, 8 to 12 shoreside.
They're chatting away on the other channel. They might not be with you for another minute. All I can say.
We'll just keep heading this way then. Easy peasy.
Hey, Jen, can you hear me?
Okay, this is Jen. Can you hear me?
Yeah, we
Loud
can
and
hear you.
clear? Yep.
Yeah.
Okay, let me try that again.
Oh, you're off.
Here's
You're
Jen.
off SPL, Bob.
I guess you can't hear me.
There we go. Now we can.
All right, so I heard Kurt. This is, can you hear me, Jen? You can hear me, Kurt? Can the van hear me?
We can hear Jen. We can't hear Kurt.
All right.
Can you hear Kurt
do you
now?
have-
We can hear Kurt now, yes.
All right, the
Hi,
gang's
Sam.
all here.
Ooh, hello.
Nice to join you all
Hello, 8
tonight.
to 12.
Yes, nice to have you here. Understand we are completing a transect.
Yes, that is the rumor.
Perfect.
And from what I understand, Sam, we are pretty close to the end.
What was your target distance? 200? 250?
200, I believe.
Ah, then we are very much so near the end. Looks like we've just gone over it, so we will complete this transect. It probably happened during the watch change. Perfect.
It's always a convenient time.
Easy peasy. All right, we'll just make it there. And the ship is stationary, so we can look around here or continue on.
Yeah, let's search around, kind of in the general direction of the next waypoint. But yeah, let's see what we can find.
Roger. Pilots, the next waypoint is in the same heading.
Okay.
So let's look around a little bit.
What are we looking for?
That's a great question. Science, what are we looking for?
Ooh, how about a cool fish?
A cool
Okay.
fish. Like that one?
Like that one?
Like that one. But I ordered up a cool fish. Here
Done.
it
What's
is.
next?
That's a beautiful,
Zoom in now?
beautiful Pacific flatnose.
Okay.
Zoom in now?
Pacific flatnose.
I think I heard you say zoom in, Bob.
I did.
You did.
You didn't hear me?
Shall I take a picture of why?
No.
I'm going to put on the nostril cam if you don't fix that. See, channel 3, pilot cam.
I guess I gotta actually fly here too.
I wish I knew more about Pacific flatnose, but they are common at these deeper depths. Relatively deeper
Kill the
depths, at
lasers.
least.
You don't forget.
Good capture, at least in this angle.
2%, that's too much.
And also, Daniel Geiger on the science chat would like to get a zoom in of some of these lighter colored spheres in between some of the rocks, see if they are brachiopods. See one on either side of the Pacific flatnose there. Maybe on the right, there was one on the right side, and there's some others around as well.
So in this shot, you can see that that dorsal fin has a really long ray on it that's sticking up when you get just the right view. Nice.
Yeah, and similarly to their, I can't remember their forward of the pectoral fins, but there's two very long fin rays that come down off the ventral side of the body as well.
Oh, are those the anal fins, but just like really far forward? Oh no, those would be pelvic fins, wouldn't they? That's right, because they're paired. There we go. We just got here. We're getting on our science hats.
We do still have questions coming in from the public, and we're only going to be answering questions for about 25 more minutes.
Also, captures.
And I have a quick one here.
Sure.
For you. It is, what are, I'm going to read it verbatim. What are those white worm-like thingies scattered about? Love this question.
So let's see. I'm looking because I see a bunch of brittle stars and then I see little white dots on the rocks. So I'm wondering if they're looking at brittle
the
stars.
larger sea cucumbers? Yeah. That's what
Oh, the sea cucumbers? Oh, okay. I was looking for little, but yeah, you're right. It might be the sea cucumbers.
Steve,
which right now is kind of in the middle of the view.
Is it a good time for Tele?
Oh yeah, I can put it up.
I'm going to put that up.
Are you going to put the telestrator on channel 3 now?
Yeah, we can do that.
There it is.
So I was circling the sea cucumber there. I think that's what they were talking about.
Yeah, so if you're watching channel 3 of the quad video, it's a fun new tool this year called the telestrator. You can see that Steve on board sitting in the data logger chair circling the sea cucumber for us. That's so great. Thank you.
He's having a lot of fun with it.
Well, you gave people time to change channels, which, oh, there's a second sea cucumber
And
that he's
Several.
circling.
I'm just enjoying the
Great.
artwork back here.
zooming out.
Okay.
Steve,
what
how about
else?
finding one of those white spheres between the rocks? That might be a brachiopod to
Yeah,
circle as well.
I'll keep an eye out. I didn't see anything that looks
Okay.
obviously like a
Lasers back on.
brachiopod, but can we maybe we can zoom in
Okay.
on some of the
zoom
white
in now.
white balls that are in between the rocks, white spheres,
You're really
shaped objects.
I don't know. The mic's not that far. I don't understand.
Yeah.
Got some acorn worms too.
Oh yeah,
Yeah, so I can circle that one also.
the acorn
So-
worm.
Any of these?
Are you talking about the, like, zoom in more out?
Yeah, let's just take a peek. So this looks like, what is that? Can we go in? Snail.
Yeah.
Pan
Yeah.
right. I thought I saw something else. Oh,
Zoom
a little
out.
too
Yeah,
far.
snail.
Right. In there?
Nope,
Uh,
it was nothing.
Okay. We'll keep our eye out for them though.
I don't see anything that looks
And
obviously
Daniel,
like a brachiopod, but I'll keep my eyes out.
Okay, great.
Sounds good.
Can you
In
get
general.
a junk
Yeah,
zoom?
appreciate that.
Science,
have
we want to
another
keep
really good
moving here?
I think that sounds like a good idea, Sam.
Roger. Bridge nav.
Sam, at this depth, what's the delay going to be between a ship move and Argus moving, do you think?
That is unacceptable.
It's a weird thing.
We zoom in again now? Starfish?
No, it went off screen. It was a floating thing.
Oh, floating thing.
That is a brittle star that we can see there.
It's out of view now. I think it
The
was
bright
floating down
orange.
current, so not worth
Okay.
chasing.
Bob, do we usually fly or do these transects with the camera sled extended or retracted? Seems like I'm zooming a lot to get past Mongo.
It's all the way out.
Oh,
All the way out.
okay.
That's coming in.
No, don't want that.
Oh, that's all you get.
Just being picky.
Yeah, you are.
Is Mongo tilted, like rotated inwards?
Nope. Mongo is totally as outboard as it can get.
All right, let me see.
We have a really good fish adaptation question, and it is, do deep sea fish use their eyes to see or sensors in their skin?
That's a good question.
What was the last? Yeah. What was the last part of what you said, Claire?
I'll repeat it one more time. Do deep sea fish use eyes to see or sensors in their skin?
Well, yeah, the
First,
main,
Kurt?
so there's not a lot of light down here at 1,550 meters. Very almost probably zero light, except for when a large ROV comes down to explore around. A lot of ambient light, that is. So yeah, most fish have what's called a lateral line. That's an organ that runs the length of both sides of both of their sides, and that can sense pressure and in some fish electrical signals. So any changes in water movement or things they can sense. And it's a very well adapted organ. Less familiar with what fish eyes function down here. I'm not a fish biologist, unfortunately.
Well,
I
Kurt,
will
if I
look
could
that up.
add to that, though, there is light that's produced by other animals down in the deep sea. Even when we don't have sunlight, quite a wide variety of animals that live outside of the zone that's influenced by the sun can produce their own light. It's called bioluminescence. And so some of the animals use it to communicate with others of their own species, like to find a mate, while others might use it to attract prey
Yeah,
or
it's pretty
to
bad.
scare off a predator.
Yes, exactly. Thanks, Jen, for adding that.
Sam, was my audio bad, or is that something else?
Yeah.
Sorry,
it's-
Jen. Yeah.
Well, I'm working on that.
It is?
Yeah, it looks like Al's doing
Okay.
some troubleshooting.
You're size C, correct?
Okay. Yeah. No, lead C.
Lead C, I'm sorry. Lead C.
Thank you. Okay. Thanks, Al.
My pleasure.
Yes, and to add to what Jen said, if everybody heard that, because of the low light levels that, the light levels are coming from other creatures down there and that are bioluminescing, you often notice that deep sea fishes have quite larger eyes than their shallower counterparts. Not always, but that's kind of a typical characteristic of deep sea fishes.
You zoom in there, Al?
Yep.
Okay.
There's a
zoom
kite
out.
in there.
Yeah.
Could be confused for
Oh,
a
yeah.
brachiopod, I suppose.
Yeah.
I'll try and circle
Especially
it.
for those that don't know what they're looking at. Oh, you zoom out?
And thanks to our scientists ashore for adding that, some of the context for that question.
Yeah, for is my audio okay right now? I restarted it, hoping it might get better.
It is a little bit better, but it can be improved more. I'm trying to make changes here, but my computer's not cooperating.
Oh.
See, you're not alone.
I'll just pitch it up to Kurt because his audio is better, but maybe you could help explain to folks who may not be familiar what Scientists Ashore is and how we're being helped by a big group of very smart people.
Sure, yeah. So the unique opportunity that Nautilus provides with satellite capabilities is that we're streaming live video and audio up to a satellite that, as I understand it, the satellite feed comes to a receiving station at the University of Rhode Island Interspace Center, and then that is being broadcast live over the internet to all parts of the world, so everybody can join us, and it provides
Can we zoom on the
great
octopus?
opportunity.
You zoom in, Al?
Yep, zooming.
Oh, yeah.
Sorry to interrupt, but an octopus.
No, that's a worthy interruption.
Required. It was like glamour shot, please. Oh, it's so cute and little.
It
Speaking of big eyes, huh?
looks like the Who Me look.
That's great. Nicely it blends in too.
Mm-hmm. I love the texture on the skin. Really almost mimics the rocks.
Mm-hmm. Di is pointing out the little, maybe the little white stick thing to the just below it, and she thought it might be a coral. It could be a hydroid too.
Gonna get moving. So one thing I've learned that some octopus have 2 rows of suckers on their arms and others have 1 row, and this looks like it has just 1 row. And that can help us tell which species it is.
That's right, and Erica Frew in the chat has suggested it might be a granulodone, if I'm pronouncing it correctly. I forget.
After we're done with that, could you have any more zoom to look at that coral, or are we out of?
We're out of lens.
Okay.
Steve and his coral.
Steve, are you? I was about to ask you where it was, and he used the telestrator. Boom, just like that.
It's worth every penny.
What do you think? What kind of coral do you think that is?
Yeah, I was hoping to
get a venture a guess.
I think it's just so small. I think it's
pretty dinky, yeah.
Yeah,
I think it's far out in front.
It's probably a small primnoid or a hydroid. but I'm going to bet small primnoid, seeing that the polyps are paired. That narrows it down to a couple of different genera, but that's the best we can do for now.
Hey,
All
Sam,
right.
is the ship still moving?
Nope, we're stopped.
We're right dead screen and
Yeah.
overhead.
Okay, all set with captures.
Okay. Moving on.
Oh,
Where's he going?
That sounds great. Oh yeah, that's a nice view.
So we're not moving on.
It's putting on a show for us at this point, he or she.
We have lots of questions about the octopus. I'm just going to pick one of them. It
Okay.
is, why do the octopus like to live on the seafloor?
Well, not all octopus do, but this kind of octopus is a benthic octopus. And if you can see with the coloration that the habitat on the seafloor provides places to hide, but also lots of different animals to eat. So let me see, there actually is one of our scientists ashore is giving us some great information about this particular octopus. It's saying that this is Grandolodoni borapacifica, and things we've already noticed, it has one row of suckers. The skin is tuberculate, which means kind of warty or granular.
Also,
And they primarily move by crawling over the substrate. They are capable of swimming by jet propulsion, kind of pumping their arms and like flaring their webs and that kind of thing, but it's not that's more of their getaway type of movement versus what we've been seeing
I think
here.
we do have a brachiopod actually, far left of the screen.
Look at that. This lovely octopus was helping show us the way, right? Like over here.
So I'm thinking
And then,
right here on the top of the
yeah,
hilltop.
doesn't say anything about what they eat, but maybe this octopus eats things like little crustaceans or little shelled animals, like
Circling
this
at channel
brachiopod or
3 here.
snail, that kind of thing.
Do we want the brachiopod?
do you have any more zoom?
Nope.
It's really small and flat.
Really tiny.
against the
Everybody
rock.
wants more zoom. I have no more zoom. More zoom for you.
You're gonna need to
Yeah,
attach
Bob,
that
I'm kind of waiting for
telescopic
Daniel
lens.
to chime in to see if he wants us to collect this.
Chin extender.
Yeah, it's so small.
It's pretty,
yeah.
It's not very far out in front of us here. Zoom out, Al. You can see.
That's different.
See?
Yeah.
For
Oh,
scale.
wow.
Yeah, you're totally,
Oh,
that's gonna come up in
very
pieces
small.
for sure.
Yeah, you can zoom in again, Al.
Jen, we're really losing you.
Yeah, I'm trying to work on that, but there's a problem here. I'm trying to get help with the computer. When I need to spin my wheel to scroll for the window I have. Well, it only changes size. It gets so small I can't see. Somebody changed it. I'm trying to find out who did so we can change it
Oh
back.
gosh.
Just conferring back.
So, yep, Daniel asked for a collection, if possible.
Okay.
Is that a slurp?
It's not a grab.
Pilot challenge number 1.
Yeah, right.
Unless you want the rock. I guess we can get a rock.
That's an idea.
What's your thoughts?
I think we got to poke it to know. I don't know how much that rock is attached. It looks like an attachy kind of rock.
I like that idea, Bob, to see if the rock is loose.